Welcome to our Bioimage Informatics Facility webinar on TissUUmaps 3. So TissUUmaps is a browser-based tool for GPU-accelerated visualization and interaction exploration of tens of millions of data points that can be any kind of points you want, and that overlay tissue samples or other kind of images. With TissUUmaps, the users can visualize markers and regions. They can explore spatial statistics, quantitative analysis of tissue morphology, and so on. They can also assess the quality of, for example, in situ transcriptomics data decoding. And you can display multi-resolution images. You can customize, share your images and points, and you can also integrate it in Jupyter Notebooks, or interface with other tools like Napari or ScanPy in Python, or many other kind of tools. Yes, so that's a short introduction. And what is the challenge with TissUUmaps? Well, we want to view and share images that can be quite huge. They can be in like 10,000 pixels width or 100,000 pixel width. And then we want to be able to zoom in these images. So we want to have access to the pixel scale information so that we can see like subcellular resolution, but we want also to have a very fast overview of the image. And we want on top of that to display our markers and the same at every scale and without any lag or like processing time. So yes, that was a short introduction. So today we will start with TissUUmaps installation. How can you have it on your system locally? How can you load your images, your marker files that are CSV files, so it's comma separated value files. How do you change the display settings, display different kind of markers, pie charts, and so on. How you play with regions also. And then how do you make projects from your files and share them online on different kind of servers. So first, if you want to install TissUUmaps locally, you can. We have a standalone application that you can download from our webpage. So here is our webpage, tissuumaps.github.io. And in that webpage, you have a download folder here. And in this download page, you will have different ways of installing TissUUmaps. The first one is for Windows, and it's the fastest. If you have Windows, you're lucky. You can just download the installer and follow the instructions, and it will install it for all users on your computer or for just you. If you don't have Windows, if you have Linux or Mac, then you can, or if you have Windows but you want to install it like in Python, then you can follow the pip installation that is like Python based. You will create a conda environment and then install TissUUmaps, and it will take care of all dependencies. So basically, you just create a TissUUmaps environment in conda. It will download dependencies that are from conda. That includes VIPs, which is quite difficult to install if you don't use conda, because you have to install it manually. So I would strongly suggest that you install with the conda environment. Once the conda environment is installed, you just have to activate it and then install through pip TissUUmaps. So you just follow the instructions here, and then you can run it with the TissUUmaps command. Or if you installed it with the installer, you can run the program as a normal program from Windows. Okay, today we will play a bit with some data. So we will start with raw images and CSV files. I took the data from the Human Developmental Cell Atlas. Actually, TissUUmaps is already the tool that is used by the Human Developmental Cell Atlas to display their data online for sharing when they have publication. So you can have a look at their webpage. And today we will play with the lung tissue data. That is in the paper Developmental Origins of Cell Heterogeneity in the Human Lung from Santolidis and al. So if you follow this link here, yeah, that's the paper. And you can also have a look at the Human Developmental Cell Atlas page, which is using TissUUmaps and describe all the tissue types. I can put that in the chat so that you can access it if you want to play with it later. And today we will play with PCW6, so after 6 weeks, and the data looks like this. And we will see how to create basically this page when we have only the raw data. So let's start with this. We have a TIFF file that contains the image here, and we can look at the properties of the file, and we'll see that it's 13,232 pixels on 9,649 pixels. So it's a quite big image. It's not huge, but it's already quite big. And then we have gene counts and cell types and a dictionary of colors for our cells. I will start with the gene counts.csv. It's actually a CSV file is just values separated with commas, so you can open them in Excel, and then you see what you have. So here we have 3 columns. We have X and Y coordinates of our points and gene name. So it's a very, very simple file, and we see that we have 494,150 markers, so different points, and we want to display these genes on top of our image. So I will start by launching TissUUmaps. So you can, if you have it installed locally on your computer, you can start with the button, or else you start it from the command line with TissUUmaps. So here we have the TissUUmaps interface. There is nothing loaded right now. You could open an image and like go to our website to have some help. We are running version 3.0.6. You can check that you are up to date. And then I want to load my image. So the fastest is actually just to drag and drop the image. And you see that even though it's a quite big image of 10,000 points on 10,000, it's actually very fast to just display your image in TissUUmaps because it's pyramidal files. So it will load the image instantly if it's in the correct format. If it's not in the correct format, and I will come back later on the format that we support, then it will convert it automatically and try its best to make something that looks okay. So that is for the image, and we can play a bit with the image layer settings here. We can like toggle the image on and off. We can change the opacity of our image. We could load other image as different layers if we had, for example, different staining or segmentation or anything in image format. We can also play with saturation, but on the black and white image, it will not change much. We can change the brightness and so on. So we have some filters that we can apply. You have actually more filters than you can select if you want to change more settings. Then we want to display markers on top of that. And we say that we will start with the gene counts. So I will do the same. I will just drag and drop the CSV file here. And you see that it creates a new tab with my CSV file and settings. So the tab name, I can rename it if I want. For example, I can just call it gene counts. Then I will load my X and Y coordinates, and that will directly come from our CSV header. So the column names in the CSV file. And then I can actually directly display the markers here. And you will see that, well, it doesn't look very nice for multiple reasons. First, the markers are really big, so I can reduce the size. And then they're all the same color, and they're all in the same group. So we will go to the render options group here, and we will select group by gene. So then each different gene will be in a different line and in a different color. And then we can update the view, and directly we have our list of genes here in alphabetical order, and we see that we have quite many. And for each gene, we have how many points we have of that kind, the shape of that marker, and the color. So if we and you can actually sort either by gene or by count. So if I sort by count here, I will have like the highest. Well, that is a bit strange, but yeah, you should be able to sort by count. And then here, if I display this, I will see like only this kind of gene, or only this one, or only this one. So, and you can play with, for example, if I want to display this specific gene here with other shape, I can change it to disk, and then they will have a disk. I can change also the color and put blue, and then they will be all these kind of genes will be blue. So that's the basic settings. It's very fast to display any CSV file with any settings you want. And you can actually add more information. For example, if we had like barcode for each gene type, we could display it as an extra column here. Or we could play with the colors, put random colors for every gene, and then it will like select different kind of colors. Or we could like use a dictionary that use one specific color for each gene, and we could display our dictionary. But for that, I will actually look at the other data set we have here, which is the cell types. So from this set of genes, there was a segmentation of each cell that was done on the image, and then looked at local information of the genes, or like the genes inside each cell, and then clustered that into cell types. So in this file, we see that we have way more columns, way more information. We have like an index here. We have X and Y coordinates. We have a cell type that is a string with like different names for each cell. We have clustering with latent. We have the number of counts inside the cell. And we have the UMAP coordinates 1 and 2 on UMAP, 2-dimension UMAP projection of our data inside the cells. We have also one example of cell count for the inside these cells. And then we have probability of being of each cell type. So that's like the maximum cell type probability. And here we have the exact probability for each cell type. And we have quite many cell types. So we will try to load this file now inside TissUUmaps. So I will do the same as before. I will just drag and drop here. And now it's loaded. I can hide the genes and just go through cell types. So here, I will just call this tab cell types. I will select the X and Y coordinates, and I will group by cell type. And then I have my cells here. The first thing we can see is that actually we may want different scale for our genes and for our cell type, because the cells are way bigger. So I will go back to 100 here, and oops, sorry. Yes, and what I will do is I will make the genes smaller. So I will go in advanced option, and I will put a size factor for the gene tab here of 0.1. And then you will see that we will have the cell types at normal scale and the genes under with lower scale, so that you can visualize both at the same time. Okay, and then I will also hide the genes for now and play with the cell types. So what can I do more? Well, I'm not very happy with the colors, because the colors doesn't fit the colors that were used in the publication. So I want to use the exact same colors as the one that were used during the publication. So for that, I will use what we looked before. I will color by group, and I will use a color from dictionary. And luckily, I have a dictionary here with each cell type name associated with a RGB color. So I can just copy that and put it here, and then update the view. And then we have the correct colors that correspond to our paper. And then if you want to publish that, it's better to have the same. And okay, so now we have something we are quite happy with. I will show you some more advanced options, but first I want to save these inside a project, right? So I want to save both the gene counts and the cell types. I will start with the gene counts, and I will save it in a button. So TissUUmaps work with buttons. So basically, if you want to have a file saved inside your project, you have to generate a button from your tab. This is the path to the CSV file, and that's quite important to keep so that it knows where to download it. And here you can change what the button will say. So for example, here you can say display gene counts, and you don't need a comment, so you can just add your button. And then you will have a new button here. And for example, if I remove my tab now, and I just hide the cell types, if I click here, it will recreate the tab with all the settings that were saved. All the colors, the shapes, the counts, the gene names, everything. I will do the same for the cell types here. I will generate a button from that, and I will call it display cell types. And yeah, now we have two buttons. I can remove all my tabs, and I can just, if I want to have the cell types, I can load my cell types here, and they are with the correct colors, correct shape, everything. Okay, and now I want to save the whole project. So I will just save the project here, and I will go to the folder that I have here, and I will go in the same folder as the rest, and I will call that my project, or maybe something more, we can say PCW06 lung. The projects are in TMAP format, and save. Now, if we go back to the folder, we have a new file called lungpcw06.tmap, and that contains all this project. So if now I can close TissUUmaps, I can actually start it directly from the new file we created. It will start the TissUUmaps standalone program. You have your image with all the image layer settings you choose, and you have your two buttons. If you want to display cell types, you can, and they will be loaded. We can have a very fast look at what does that look like. And it's actually a JSON file with the different like information of your project as settings. And we can see the marker files here. That's our buttons with the path and some other tools. So some other settings. So here you can change manually some settings that you cannot change from TissUUmaps. For example, we can ask it to auto load on program start here. And so if we load, let's say, the cell types here, and I save that, and then I go back to my program. When I start here, if I open back, I can also just move it here. Now you see that it will preload the cell types when you open, and it will remove the button. So the button has been transformed directly into a tab. We don't want to do that automatically because sometimes the CSV can be quite huge, like hundreds of megabytes. And if you share that online, you don't want to force people to download all the CSV files by default. So that's why we go with the button so that people can select the files they want to display when they open the image. You have a region tabs, and here you can actually draw region or import regions from other tool. It imports in the GeoJSON format, which is the format that is used by many other tools like that. For example, QPath can export in GeoJSON. So if you have a segmentation of cells or of objects or anything or in QPath, you can load them in TissUUmaps. I can show you how it works. So if I draw a region here, I will have like a new region class here, and let's say that this is just normal tissue. I will have like the tissue tab with all my tissue regions, and this one is region 1. Then you can analyze what's inside of this. And here we have like all these cells. So we have 55 cells of airway smooth muscle. Okay. And we have like 21. So we don't have a second name with columns, so it's all undefined. We just have like the main cell type. But yes, you have everything here. You can also then export these regions with what they contain as CSV file. And the CSV file will contain for each region the markers inside this region with the region name and class and everything. So you can do more analysis after with your CSV file. But we don't do any of the analysis directly in TissUUmaps. I will show you a bit later. You have plugins that you can use, and there you could make plugin to do some more advanced analysis, but it's not what it's meant to be. It's really just for visualizing locally on your computer and sharing to other. So there was one question about how to create the cell types text file. Do you mean the cell types like the CSV file here? So this here, it's you have many tools that can create that. I don't know what they use here, but most of the time you use R or Python. So in Python, you have the Pandas plugin module that can create data frames that looks like that. And then you can export data frames to CSV. And it's very similar to what you can do with R. And yes, I wanted to show you one more thing here in the advanced option that I didn't talk before, and that is pie charts. So let's say that instead of displaying the main cell type here, I want to display the probability of being of each cell type. And if we go back to the Excel file of cell types, we actually had all probabilities for the different cell types. So we had like many individual columns here for each cell type with the corresponding probability for each row. So what I did here is that I combined that into one new column where I put all the cells names, and then each row is combining like with semicolons all the probabilities. Once you have a row like that, a column like that, you can load that into pie charts directly in TissUUmaps. So I will select this last column that I've created, and I will update the view. And then here you have the list of pie chart, the list of cell types, and your pie charts. Of course, here it's using a default color space, which is not really interesting because it only have like 10 colors. So it's looping through the colors. So again, we will take our dictionary from here and we will put that in the pie chart colors. And now we have the same colors for the cell types in the pie charts as we had without the pie charts. So if I toggle in and on the pie charts, for example, I can go back to the main cell types, and I can see that it fits the cell type, the main sector of the pie charts. I can also select a fixed shape here because I'm not happy with all the different shapes. So I want to use a disk everywhere and then update. And then, yeah, so now we have disk everywhere. And if we use pie chart, we will see that it just converts to the pie charts. This gives a bit more information, and you can fill anything in your pie chart as long as you have it in one column of your CSV file. To continue through these advanced options, you have the marker opacity here, so you can change the opacity of all your markers so that they're a bit transparent if you want to display things under it. And I will remove the pie chart, and you can select also a thing to display when you click on the marker. So right now it displays like the main cell type when you click on it because it's the grouping key. But you can display more information. So we have, like, for example, if you want to display the tab name, so like the cell types here, you will display tab, and maybe you still want the key to be displayed, so you display the key. and maybe you want to display the cell counts inside. So we have a column in the CSV that is called counts, I think. I will just check that it's the correct name. Yes, we have counts here with the number of counts per cells. So if I select like call counts, then I will have that, and I can add counts after so that we know what we're talking about. So now if I click here, I will have, it's in the tab called cell types. It's proliferating epithelial 2, and it has 162 counts. And for each marker, you have a different value. So here we had markers with cell types, and we used the coordinates X and Y that are spatial coordinates so that we have it on top. But we actually developed a feature space plugin here. And so when you load a plugin, it goes in the new tab plugins, and then you have like your settings for this plugin. So we have the feature space plugin where you can select one data set. So here we have the cell type data set, and then you can select which feature space you want to display. So we will display UMAP 1 and UMAP 2, and then you can click here. And then you will have next to your spatial coordinates image, you will have the UMAP coordinate image. You could also just load the UMAP as one program. In TissUUmaps, you don't need to have a background image. So for example, if I go back to my folder here, and I start a new instance of TissUUmaps, I could directly take the cell types here, copy that, and instead of displaying X and Y, I could display, where is it, UMAP 1 and UMAP 2, and group by cell type. And then you would have your UMAP coordinates directly in TissUUmaps. But to go back to the plugin that we have here, the nice thing is that now the two are correlated. So if you go back to the markers, and let's say you want to look at one marker type at a time, you can like go through your classes here, and you will see on the right side and on the left side where each one fits. I will put that a bit bigger so that it's a bit more visible, and I will reduce the opacity of the background. Yes. So here you can see how it's correlated. You can also select a region in the UMAP space. For example, if I select this thing and ask, where is that? It will display only the cells that are in this region in the spatial coordinates. And you see, like, yeah, these are just around these glands, while if I select these here, they are a bit outside. So then you can compare, why is the UMAP coming from, where is this area, or where is this one, for example, and you see that it's all around, or this side, or this side. So it's very interactive. You can play with your data. You can check what you have in the different space. It can also give you different ideas of visualization. For example, here we see that we have quite different things in the top and in the bottom of UMAP. So we could think like, oh, I want to change the colors. So I instead of color by marker type, so by cell type, I want to color each marker depending on some value. So I will color by marker here, and I will select the Y coordinates of the UMAP, so UMAP 2. And then I can, so this is a value from 0 to something like 0 to 5,000 or something. And then I want to select a color map so that it knows how to color that. And then I update the view. And then you see that it will take the Y coordinates from 0 to the maximum and put that in a color map from, like here I chose the turbo color map, but you can change to any color map. And then it will change on the right side and on the left side, so that you can see, okay, you really have a strong contrast between the cells here and then around and then the green. So it's really informative to look at that. And in the same way, you can select things in the UMAP coordinates. And how do you share a project that is created in TissUUmaps online? You have two ways. The first one is if you have a Docker container running TissUUmaps software. And that is a bit technical because you need a system engineer or someone to install it. And usually it's the TissUUmaps Docker image will run on a Kubernetes engine. So it's quite big and difficult to, know how to install everything and make it secure. But that's what we have running in SciLifeLab SE platform here. And that's what we are using for the Human Developmental Cell Atlas. So if you go to this link here, you have an instance of TissUUmaps running. And for example, if you go to the Human Developmental Cell Atlas here, you will see that all of these are run directly on the TissUUmaps instance. So then you don't have to create static web page or anything. You just push your project on the platform with your images and CSV files and TMAP files, and then it will load it here. But it's a bit technical, so it's not the easiest. So what you can also do is to push a TissUUmaps project on a web server, for example, Apache or Nginx. That's usually what you have when you have personal pages at your university, or yes, you have a web server that you can run, that you can put index HTML or any files and access them. But that needs a bit of more processing because you want everything to be in a format that can be read by your browser. So for that, we have an option here to export your project to a static web page that you can push on a web browser, on a web server. So I will go here and I will select a new folder that I will call, for example, static, and then I will save everything inside this. So it will say loading, exporting to static web page. That can take a bit of time because what it creates in this static folder is new data for the image especially. And this will create your image in a format that is called DZI, that is actually a pyramidal format with all the tiles as different images. So if we go, if we go into the image folder here, we see that we have different scale. And for each scale, we have the image at a different size with tiles. So here we have all the tiles, and that can be loaded in the browser. We also have like the CSV file saved. And then in the main folder, we have our indexed HTML and all the dependencies that are needed for putting the project online. So just, and here it says that exporting is done. So just by creating that, you will have something that you can share on your server. And so here, for example, we have a server running here. And so I will put my static page to my web Apache folder here that is running. And then you can open it in your browser. So for example, I have the static folder that I just created here. And in this static folder, I have my TMAP project that can load, and it contains everything that you had in the standalone application. It's exactly the same as it's all web-based. You can also display the gene counts on top of your cell types. So you can click on a gene count, or you can have the cell types here, and you see that it saved all the settings that I had. Here display like proliferating epithelial 2 with 133 counts and so on. You can also change the settings from here. So if I want to display, let's say, pie charts instead, I can just click on pie charts and it remembers the settings that we used for the pie charts. And then here you will have in the browser something that everybody can access and with your complete file. So about the image format, question that comes often is my image doesn't load, or it's all black in TissUUmaps, or it has a bad intensity range. So actually, TissUUmaps loads images that are in pyramidal format, but also that are encoded in 8-bit. 8-bit images means that each pixel is encoded in a range from 0 to 255. And that's enough for visualization. So that's what we use in browser to display images, because your monitor only displays 255 intensity values for each pixel anyway. The problem is that some images that you acquire with microscopes or any tool or any hardware, they are usually in way more, they have more information per pixel. They are encoded in 16 bits or 32 bits, which means that they have values that are in bigger range than from 0 to 255. So if you load that kind of image into TissUUmaps, it will automatically rescale it to 8-bit. It will try to do that in a smart way. So it will like remove outliers and keep the main range that is used in your image, and then convert that into 8-bit and then display it. But sometimes it can fail. It can fail to find the correct range, or your format is not recognized by TissUUmaps at all. And TissUUmaps is using VIP's library to convert it, so some images are just not supported. So the best thing is to use other tools that are good in converting images. For example, you have bioformat. You have, like in ImageJ, you can load your image and export it into a format like TIFF in 8-bit, so that you can choose your range as you like it to display it in TissUUmaps. Another thing is that the file needs to be pyramidal, so that it's fast, to go from the very zoomed image to the unzoomed one. So pyramidal means that inside your TIFF image, all these different scales are saved as different layers. And then when I access one point, instead of loading the whole image, it will just access the correct scale and the correct position of the tiles, and then just send these. So TissUUmaps, if you load an image that is not pyramidal, it will have a conversion tool that will convert it into pyramidal. So let's say that I want to load a JPEG image here. I don't have a JPEG image, but yeah, I could open that here with, let's say, with GIMP and save it as a JPEG. So if you load a JPEG image into TissUUmaps, it will try to convert it into a pyramidal format automatically. So it will create your new TIFF file, and it will put it in a folder called TissUUmaps. So we can do some example here. If I export that into, let's say, this .jpeg here, export. And yes, export. So now I have a file that is not possible to open directly with TissUUmaps, right? So if I open back an instance of TissUUmaps and I close that, and now I want to open this JPEG into TissUUmaps, it will not know how to open this image. So it will create a .TissUUmaps folder, and inside this .TissUUmaps, you have a TIFF file that was automatically converted from the JPEG one. And depending on the size of your input image, of course, it can take more time to convert it, but this image is now pyramidal, and it's like it doesn't tell you that it converted it, but you can see that it has this .TissUUmaps folder with your high dimension pyramidal image. So in theory, TissUUmaps can open almost any kind of format. It has to be supported by the VIP's library, but it's quite a large range. Unfortunately, there are some new format every week or month that are not supported. For example, I know that Saga, that should be here today, I don't know if she joined, but she's working into loading bioformat images, and that's a bit tricky, and some formats like Leica are not supported and so on. So if your image doesn't load in TissUUmaps, try to convert it to TIFF with another tool, or sometimes the software coming with your scanner has an option to export into TIFF, and TIFF is always better because TIFF can be converted into pyramidal TIFF, and then you can load that into TissUUmaps. So I had just a couple more of like frequently asked questions here. One is sometimes you create a button and then when you click on it, it doesn't load anything. It's a bit tricky because the buttons that you create here, they have to know where the CSV file is and when you load it in the browser, sometimes it doesn't know where it is on your computer. It can come from any other browser folder. So make sure when you create it that they are either all in the same folder or in a folder that is accessible locally, like with a relative path. And then check in your TMAP file in the code that the path is the correct one relative to the TMAP project. So here they are in the same file. So we just put point slash, but it could be in a parent folder or in subfolder, and then you would need to specify that in the TMAP file. So always be careful. If your button or input here doesn't load the file and it's stuck in loading, that means it didn't file the CSV. So make sure that the path is correct inside of the text TMAP file. And the last one is if you create a static web page and then you cannot load it in your browser, it's because you need to have a web browser, a web server running like Apache or something like that. So be sure that you have one. If you don't, you can create a Python server. So if you open terminal here and you have Python running, you can create a minimal Apache server by just calling HTTP server module. So for example, here, if I call that, it will start a new server running locally, and then I can open it and access my file. But be sure that you don't directly open the static image into your browser like that, because if you do that, then it will try to load it locally and it will not have access to the data. I will just say thank you to everybody that contributed to the project, and there are many. From Kalina's Velbis lab, TissUUmaps is a project that was started about, I don't know how many years ago, 6, 7, 8 years ago, something like that, maybe more. And many people collaborated to the project. So it's yeah, it's a very long-term project. And if you have any issues when running TissUUmaps, you can always put open issues on the GitHub page. So you have the GitHub of TissUUmaps. It's just like TissUUmaps/TissUUmaps is the main repo. And then you can open new issues and we'll try to solve them together. Also, if you have feature requests. It's always interesting to know what people need and what people use.